let's look at some nutrition terms. An essential amino acid is one you must get in your diet. The word essential for a dietitian means you need it in your diet. The reason that you need to get these in your diet is because you need all 20 amino acids. You need access to them, a supply of them, in order to make your proteins. In other words, to make your insulin, your hemoglobin, your receptors, some of your hormones. Anyway, to get those 20, some of them you can make. You just take the ones that you do eat, and you have enzymes to rearrange them and break them apart and make other amino acids that you need. But there are some that you don't have the enzymes to do that, and so those you have to get in your diet. Otherwise, you can't make those proteins correctly. A complete protein is a protein that contains all of the essential amino acids. So the common example of a complete protein is egg white. Typically, animal proteins are more complete than plant proteins. So if you are someone that does not eat any animal proteins, you need to know what types of plants to eat together. For example, grains and legumes are complementary. Grains provide certain amino acids but not others, and legumes provide the missing ones from grains and then some others. So they come together to make a complete protein, like rice and beans, or even a peanut butter and jelly sandwich. Next, let's talk about some of the assays that we're going to do on our amino acids and protein solutions. First one is biuret. Biuret has this copper ion, which you know is a light blue. You've seen it before. It's the same one that was in Benedict's reagent. It forms a purple to violet complex with the NH of the peptide bond. So you need a peptide bond in order for this to react. So in other words, a protein should test positive because a protein has peptide bonds between all of the amino acids, but an amino acid will test negative because amino acids don't have peptide bonds. Peptide bonds are the bonds between amino acids on a protein chain. So an amino acid by itself should test negative. On this test, when it forms this color, it goes from light blue, sometimes it just goes to darker blue, or just slightly more purple looking. Next, ninhydrin. Ninhydrin reacts with a free amino group. This NH2 group is called a free amino. And so the compounds that have that always are, well, except for one, we won't get into that one, are the amino acids. An amino acid has an amino group and will form that complex to give you that blue-purple, similar color. Okay, so this is a test that's used for fingerprints, like when they want to get fingerprints off of a piece of paper. You know, it's not the same way because they're not lifting oil off the paper like they do, you know, with an adhesive, and then they use a powder. It's not a different way. They spray the paper with this ninhydrin reagent, and then it may take a few hours, but the fingerprints will show up purple. It's pretty neat, actually. Let's see. Any free amino acid will do, or amino group will do. So even a compound like this will test positive, even though it's not an amino acid. We should note, though, proteins only have one of those, one free amino group, because most of them are lost in the peptide bonds, and that just having one on the chain probably would not give you a positive test. But if the amino acid, or if the protein contains the amino acid lysine, each lysine amino acid has a free amino group, and so that will allow it to test positive. It was one of the amino acid groups we looked at the beginning.